The overall goal of the experiment is the rapid identification of molecules able to inhibit the chaperone activities of HIV-1 nucleocapsid protein in vitro. NC is a small and highly basic protein with its structure characterized by a flexible N-terminal domain and by two highly conserved zinc fingers that are engaged in interaction with viral nucleic acids. NC interacts with the TAR RNA hairpin and its DNA complementary sequence CTAR and promotes the melting of their secondary structures to make them available for the annealing of the TAR-CTAR hybrid. The protocol is performed by using the apical part of TAR and CTAR sequences. When mixed together and incubated with NC, the hybrid is formed. Threading intercalators interacting with oligonucleotides inhibit the NC-catalyzed hybrid formation. The outcome of the reaction is analyzed by standard polyacrylamide gel electrophoresis. TAR and CTAR have different mobility in the gel compared to the hybrid. Increasing concentrations of threading intercalator leads to the decrease of the hybrid in parallel with the appearance of free TAR and CTAR.
The main advantage of this technique over existing protocols such as gel shift analysis or TAR annealing is that it does not require preliminary manipulation of nucleic acids such as radioactive labeling.
This method can help answer key questions in the field of drug discovery, such as HIV-1 and C inhibitors.
Generally, individuals new to this method will struggle with the overall organization of a protocol, particularly with the serial dilution of each stock, the exact incubation time, and the correct procedure to stop the